everyone, good evening. Welcome to Akash Baiju's NEET. I'm your teacher Ankita and I welcome you all in today's class where we will be discussing about the very interesting and super important topic from the NEET exam point of view, and that is genetic code. So I hope that all of you are ready for the class. Everyone, please make sure to hit the like button for the video, and if you haven't subscribed to the channel, please make sure you hit the subscribe button also. Good evening everyone, I welcome you all to the class. Hello Vispajit, hello Radhika, hello Tanu, good evening everyone, how are you all? Yes, I hope that all of you are doing good, and I hope that I'm clearly audible and visible to each one of you. Hello Himani, good evening. Okay, right, this is a part 7 everyone. We had done part 6 previously. We have covered up DNA replication, the genetic experiments that have happened, and DNA transcription. And now we will be discussing about the genetic code. Yes. Botany कब तक खत्म हो जाएगा? See, we will be trying our best to finish off the syllabus of the botany and as well as zoology, and not just the bio subject, but of course the subjects, across the other subjects also. We'll try to finish off the syllabus as soon as possible. But of course, we all know that we cannot finish it right now. Probably we will be able to finish it for sure by the month of December, right? That should be our target. Hello Roshan, hello Dr. Kritika. Yes. Zoology, I think tomorrow you have a class with Pushpendu sir. Yes. Awesome. So everyone, let's get started. I want a very quick thumbs up. I can see so many of you are here in the class. Please make sure everyone to hit the like button for the video. Right, and let's get started without taking much of a time. Yes, Anish, hello. Okay, chaliye. Okay, everyone, let's get started. We are discussing about the genetic code, and first we are starting with the central dogma. क्या था central dogma? हमने पहले पढ़ा हुआ है. आप एक बार फटाफट से रिवाइज करवाइए. Yes, बिल्कुल. We will be having a test series in between. Also, we'll have the questions for the test series also. Okay, jaldi se everyone, what is central dogma? See, आज की class is a very short class, right? It's a very easy concept, so we'll not be taking much of your time. But please make sure you jaldi se answer करो. What is central dogma? Right? Watson ने बताया था या क्रिक ने? जल्दी से. Very good, right? So of course we know that central dogma, right? Of course it's a very famous theory, we can say, जो हमें दी थी फ्रांसिस क्रिक ने, right? And he said that the process involved in the conversion of DNA into a functional product, and this is what we have, right? We have the DNA. Now DNA से क्या होगा? DNA अपनी खुद की replication कर सकता है. DNA से RNA बनेगा by the process of transcription, and from the RNA we will be able to see the formation of proteins by the process of translation. So DNA अपनी खुद की replication करेगा, DNA से हमें RNA मिलेगा और RNA से हमें proteins मिलेंगे. So replication, transcription and translation. हमने replication already पढ़ लिया है, transcription भी पढ़ लिया है. अब हम translation की तरफ आगे बढ़ रहे हैं. Clear? So this is very important. ये आप सबको आना ही चाहिए. Now based upon this, right, based upon the central dogma, we can actually talk about that how the things will be moving ahead from the RNA to the protein. Now we all know that what is RNA? Now of course when we talked about the transcription, we studied that it is a copy, right? DNA acts as a template, and what happens, the RNA is a copy. It have the sequence, right? The sequence are parallel, right? We know that this is a very important thing that we all have to understand. Now this RNA will be making the proteins that we all need in the body. So ये process बहुत important है. But इस process को समझने के लिए हमें एक चीज का समझना बहुत जरूरी है, और वो है हमारे genetic codes. Everyone, I am sure you all agree. इसके लिए हमारी बहुत जरूरत है कि हम समझें कि बहुत ही genetic codes. जेनेटिक कोड्स की ऑफ कोर्स हम आगे धीरे पढ़ाई करेंगे, समझेंगे कि क्या हुआ, कैसे हुई इनकी डिस्कवरी और क्या क्या एक्सपेरिमेंट्स करे, राइट? बट उससे पहले लेट्स टेक अ लुक, राइट, एट अ वेरी फेमस साइंटिस्ट हु कंट्रीब्यूटेड अ लॉट इन दिस फील्ड. वी हैव जॉर्ज गेमो, राइट? विल बी लुकिंग एट फ्रॉम द न्यूक्लियोटाइड, जो हमारे डीएनए टू द अमाइनो एसिड्स. वी नो दैट अमाइनो एसिड्स आर द वन दैट मेक्स द प्रोटीन. सो अब हम क्या देखेंगे, अब हम बात करेंगे साइंटिस्ट जॉर्ज गेमो के वर्क की, एंड हम समझेंगे किस तरीके से एक्चुअली कोडिंग हो रही है, राइट? एंड हाउ this messages has been transferred from RNA to the proteins. Let's talk about this, right? So we know that in the nucleotide or in the DNA, we have four important nitrogenous bases. We have in RNA, we have the uracil, adenine and cytosine. ये हमें मिला हमारे RNA से. Right, DNA हमारे पास U की जगह पे T होता है. So we know that this is the combination. So Gamow said that we all know that we have four nitrogenous bases. And approximately at that time, he said that we have 20 amino acids. Very strong guess, right? And he said 20 amino acid. Now to code, अगर हमें ये 20 amino acid बनाने हैं और हमारे पास में ये 4 nitrogenous base हैं, तो हमें इनकी कुछ ना कुछ तिगड़म बनानी पड़ेगी, कुछ ना कुछ codes बनाने पड़ेंगे, right? अगर हम एक code लेंगे तो कुछ होगा क्या? What do you think? Let's suppose if I just use G. Do you think it can code for a protein? More than that, do you think that they will be able to code for all 20 amino acid? No, right? We have 20 amino acid and we just have 4 nitrogenous bases. So we cannot go with the 1. Now what about the 2? If we take G, U, do you think we'll be able to have the combination? Again, the answer will be no. Now when we have this G, U, A, if we have this combination, right, we will be able to have 64 combinations, right? We'll be able to have 64 combinations. And on 64 may say, of course, we will be able to generate or they will be able to code for the amino acids. जो 20 हैं. Clear? So everyone, are we clear with this? So इस तरीके से he said that, he suggested this कि out of 20, right, 20 amino acids that we have, it has to be, the code that has to come should become in a triplet form. Very, very important. So after he said we have this, I'm sure all of you have seen this in your textbook also. ये थोड़ा सा interesting और थोड़ा सा tricky है, but this is very, very important. We will be looking into the detail of it. But everyone, are we clear? Now what are we doing over here? We are just starting with a scientist. We'll understand in detail. एक characteristics के बारे में भी हम पढ़ेंगे, जो features हैं of the genetic codes. We'll be looking at that also. Please make sure everyone you stay with us till the end. Now that we are clear over here, we have the 64 combinations. Now let's understand more further. So we know that we have the 3 bases for 1 amino acid, right? And we have the 64 combination. Now we'll be discussing more further about the genetic code. Now apart from the scientist George Gamow, right? We have 3 important scientists. We have these 3 important scientists, everyone, and their work. Now, इस topic में से, I would not say this topic, but in this chapter, I'm sure you know that के कम से कम 8 या 10 सवाल आते ही आते हैं. So everyone, every topic is very important, and from every topic we can have questions. So when we are discussing about the genetic code, these scientists are super, super important, right? So please make sure you put a star mark on it. Yes? Yes, everyone. Hello, hello, I can see a lot of you have joined just now. Good evening, everyone. Good evening, Shalini. Good evening, Medha. Good evening, Bhaskar. Table हमें पूरा याद नहीं करना है, कुछ याद करना वो भी हम करेंगे. Yes, Bishwajit. Yes. Meghashri. Yes. Hello, Priya. Hello. चलिए. हाँ, Bishwajit will discuss about which codon has dual function also. Yes. Sihuili. Yes. Okay. Now, everyone, let's talk about the 3 scientists and their contribution. So we have these 3 scientists. We have Hargobind Khurana, Marshall Nerberg, and we have Severo Ochau. Okay? Ochau. I hope I'm pronouncing it correct. So we'll just remember the name Severo. Okay? So we have over here. Now all of these scientists plays a very important role. First we have this, right? Ochau. So what he did, he actually made an enzyme to polymerize RNA. Can you imagine, right? RNAs, what is the meaning of polymerize? Anyone? We have discussed about the DNA replication, right? What is polymerization? Polymerization is nothing but the adding, सबको साथ में लेके आना. So the enzyme to polymerize, to combine the RNA. So what he did, he made this very, very important experiments, right? This is super important, everyone. Please do remember this. Everyone, are we clear? Yes. So what he did basically, he made an enzyme that we have enzymes for there, right? The enzymes for the polymerizing the RNA. Let me write over here. With define sequence. Right, he made it with this and define sequence in a template. Clear, everyone? Yes, very good, very good. Okay, in a defined template independent manner. So what we have an enzyme with a defined sequence. उन्होंने एक ऐसा enzyme बनाया with a defined sequence in a template independent manner without any codes, right? We have a independent sequence. Super important. Then we have Hargobind Khurana. What he did, he made an artificial mRNA with known sequence. So he made an mRNA knowing that, okay, these are the sequence I have. Okay, I have A, right? I have U, I have G, I have C. So he knew the sequence and he made this artificial mRNA. This is super important, everyone. And last but not the least, we have Marshall Nagelberg, right? And what he did, he made cell-free system with required enzymes. So he made a cell-free system where, of course, we have messenger RNA, which were making the polypeptides. And with all of these experiments, as these experiments were done again and again, they gave the various results. And based upon those results, right, what they did, they decoded the nucleotide. Everyone, are we clear? इतनी बार जब हम एक एक्सपेरिमेंट को रिपीट करेंगे, उसको एनालाइज करेंगे, उसको समझेंगे, तो हमें हिंट मिलेगी. इससे इन एक्सपेरिमेंट से पहले एटलीस्ट ये अंडरस्टैंडिंग तो थी कि ट्रिपलेट फॉर्म में आएगा, राइट? दे केम टू नो दैट इट विल कम इन द ट्रिपलेट फॉर्म. नाउ व्हाट दे वर डूइंग, दे वर ट्राइंग टू kind of analyze it and prove it that the genetic code is in a form of triplet. Everyone, are we clear? Yes, all of you, are we clear with this? Yes, quick thumbs up. Yes, Deva. Are we clear with this, everyone? Good, very good. Everyone, keep the Josh really very high. Okay, chaliye. So what we have discussed over here, Biswajit, we will discuss it. Don't worry. So we have, what are we discussing about? We are discussing about the discovery of the genetic code. So बहुत कुछ हुआ, बहुत कुछ changes हुए. 3 scientists were there, right? Severovic, what he did, he made sure, right? He made an enzyme to polymerize the RNA. RNA को जोड़ने के लिए एक enzyme आया. फिर Hargobind Khurana in his experiment made an artificial mRNA with a known sequence. He knew what the sequence of the mRNA. And of course, then we have the Nagelberg, right? Nagelberg, what he did, he made a cell-free system with the required enzyme that are really very crucial, that can actually help in the conversion of mRNA to the polypeptides. And lastly, this is how the nucleotides ka decoding was done. So this is super important and very crucial. Now let's take a look of everyone over here, right? So this is super important. We were talking about the cell-free, right? Remember this, cell-free system with the required enzymes. Here we have, see. Now we have the work of Nagelberg, right? What he did, he synthesized an mRNA. Over here we can see, right? He synthesized consisting only of poly-U. Can you see this long chain of poly-U mRNA? इन्होंने क्या किया? Right? Marshall, the scientist who was working on the genetic code, what he did? He actually made a mRNA sequence, right? A molecule which just have U. And what he did? He tried to find out that what it will be coding for. And anyone, any guesses which amino acid is this? Jaldi se please write in the comment section, tell me which amino acid is this? He studied the polypeptide chain. He studied this particular chain and found that the code U is for the, very good everyone, PHE. This is the phenylalanine amino acid. So now we know the code. So this was the first, this is very important everyone, 1 mark question, right? Which was the first amino acid that the code was discovered per se is this. So remember this, right? So एक हमें याद हो गया, 3U. Then we have one more everyone. Now we have UCU, CUC, UCU. So we have UCU. then we have UCU over here, and we have UCU over here. Can you see this? UCU is coding for serine, and CUC is coding for anyone? Leucine, right? Yes. Very good. Very good, everyone. Trick हम बिल्कुल we will have the trick, don't worry. We'll have the trick for you to remember this. I'm sure in class 11th you would have studied this, all the amino acids and the biomolecules. Right? Chaliye. So this is also important. So remember these two. Now for their work and contribution in the science, Harbind Khorana and Marshall won the Nobel Prize in Medicines. Okay? So this is how the discovery happened. The discovery is very easy. Right? The discovery is really very easy. It's not very, of course, not very complicated in the way they did the research, they did the various experiments, and that's how they got the information. Now we are clear that in genetic code we have 3 triplets, right? 3 triplets will be forming a codon, and that codon will be coding it for the 20 amino acids. And these 20 amino acids are the one that will be making the proteins. So we are now clear that when we have mRNA, which have some codes, that code will be understood. Triplets hai, right? And from there we'll be able to have the proteins. Interesting. Now everyone will be looking at the silent features of the genetic code. So let's talk about it, everyone. One of the the first important characteristic features which we all have just now discussed is that triplet codon. There are 3 nitrogenous bases that will be coming together and will be forming a codon. And this one codon will be coding it for one amino acid, right? So these codons will be coding for the amino acid. I said one because this one will be able to code for one. Okay? Clear? So this is easy. This is nothing very fancy. We all know that these 3 nitrogenous bases, right, like G, C, U, when they come together, they form the codon, and they will be coding for the amino acid. So that's one. Then, second important silent feature of it is that they are non-ambiguous. They are very clear. Ambiguous means what? Clear, clarity nahi hona. I have no idea what we'll be doing. But these codons have a lot of clarity. They have a clear-cut clarity. So the codon will always code for a specific amino acid. It's not that कि अच्छा CUG. ठीक है, आज I'll code for maybe today I'm in a mood to code for, let's say, lysine. Okay, tomorrow I can code for serine also, right? So it's not the case. They're very, very clear. So codons will always be coding for their specific amino acid. Everyone, are we clear? They will not change their mind कि अच्छा आज इसके लिए code कर लेते हैं, अच्छा आज मैं ये वाला amino acid बना तो no, that's not the case. Third very important silent feature of the genetic code is that they are continuous. Basically, there are no punctuation. They're very continuous, right? If you read it like this, we can clearly see over here. We have some gaps, but that is for our understanding to differentiate. But how we'll be reading, right? We'll be looking at in this particular way. There's no commas, there's no full stop, there's no breaks over here. Priya, yes, welcome to the class. Priya, have taken your name, Suchita. Good evening. So are we clear, everyone? So they are continuous, there are no punctuation. Clear hai? Then comes the degenerate. Now this is very tricky. This part is a little bit tricky. What it says that amino acids, sometimes some amino acids are coded by more than one codon. Let's suppose we have GUC. That can be coded, that can for valine, right? But do we have one more codon for this? Yes, right? We do have one more codon for this. So if we replace this C with an A, it will still be coding for the valine. Everyone, are we clear? क्या समझ में आया हमें कि एक amino acid को बनाने के लिए sometime we could have 2 codons. Right? We can have 2 codons. 1, 2, 3, 1, 2, 3. These codons can actually code for one amino acid. That is a possibility. Clear? Everyone, are we clear? So this is important and this is a trickier part. So I want you to understand it clearly. Chaliye. Very interesting point is that there's no overlapping. There's no overlapping. What is the meaning of overlapping? They do not overlap with each other, right? They're very, very specific. It's not that ke, acha, you know, GUC will make some, start coding for something else also, or you know, they'll start changing it. No, they will not. They're very, very specific. They will be coding for their amino acids only. So everyone, there's no overlapping in the genetic codes also. Very important thing over here is the stop signal. This is one of the important feature and very lot of questions that usually come from this particular part, right? We have the stop signal. So out of 64, 61 are active, 3 are inactive, right? The 3 codons, right, are kind of not coding for any of the amino acids. So we have 3 codons. They do not code for any of the amino acid, but they act as a stop codon. So if they are there in a sequence, right, that means that it's a stop codon. We will not, for the process that is happening will not occur further. Clear? Yes, so here we have 3, right? We can see UAG. Very good. I can see others writing UAG, UGA and UAA. Very good. Everyone see, ye yaad rahega. Clear? Yes. Very good, Shalini. Awesome. So everyone remember this also. What we have? We have UAG, right? We have UAG and UAA. These are stop codons. Stop. Everything will be stopped. So that's one of the important feature. Then we have dual nature. I think Biswajit was asking in the beginning, ma'am, what is about the dual nature? So we have a codon which is UAG, which actually codes for methionine, right? Of course, it's an amino acid. Along with that, it plays a very important role in initiating also. So it acts as an initiator codon also. If we have to start, right, this codon will be there. And further, of course, it will have its own importance role in making the protein. So it will take part there also. But this have to be there. It's a start codon. Clear? So if this is there, of course, the process will start. The protein process, the making of the proteins will start, right? The translation process will start. So we'll remember this. Okay, this is a start codon. ये आ जाएगा तो एकदम काम चालू हो जाएगा. Everyone, are we clear? See, we have discussed so many silent features. We'll come back over here again. But I hope that all of you are clear with this. Now, one of the important and the easiest feature of this genetic code is they are universal. Means that we have in the same amino acids in the living organism, right? So the codons code for the same amino acid across the living organisms and in virus, but we have some exceptions because kind of here and there in every field there will be some exceptions. So here we have some exceptions. We have the nuclear DNA and the mitochondrial DNA. So we know that in the mitochondria they have their own DNA, which means that when they have their own DNA, they have their own ribosomes also. When they have their own ribosomes, that means that they can make their own proteins. But there is a difference between the nuclear DNA and the mitochondrial DNA में codons में, right? Let's take a look over here. So, in nuclear DNA, AGA and AGG codes for amino acid, arginine, right? They are coding for this amino acid. In nuclear DNA, AGA and AGG are coding for an amino acid. Clear? But everyone, in the case of mitochondrial DNA, these two act as a stop codon. So we can clearly say that we have an exception over here. Now in the mitochondrial DNA, if this codon is there, that means stop. But if we have this in the nuclear DNA, it means that we have, basically it's an amino acid ka code. Clear? Everyone, are we clear? All of you, are we clear with this? Right? So let's quickly revise. Everyone, first of all, do take the screenshot of this. Jaldi se take a screenshot of this. Bhaskar, we are trying to have a fixed timetable. I know that there's a gap, but we will be coming regularly in the future. So yeah. Yes, we will be able to finish our syllabus hopefully by the month of December. Or before that also, let's try. Everyone take the screenshot. Jaldi say, give a quick thumbs up. Very good everyone. Chaliye. Okay. Now let's quickly revise everyone what are the things we have studied. But not from here. We'll quickly go to the NCERT. Chaliye. Let's read this NCERT, and with this we will be done with the genetic code. During replication and transcription, nucleic acid have copied to form another nucleic acid. Hence, these processes are easy to conceptualize on the basis of the complementary, right? They are complementary to each other, so we can easily understand कि हाँ इस तरीके से the process is happening. The process of translation requires a transfer of the genetic information from a polymer of nucleotide to synthesize a polymer of amino acid. So now we see a big change. From DNA to RNA is really very easy. But from the RNA to the amino acid is a little bit difficult. Why? Because from the chain of a nucleotide, we have to make the amino acids. Neither they have nothing complementary between these two. Amino acid and RNA does not have anything complementary. So how the things will work? Let's see. There exists ample evidence, supports and other things that actually help us to understand about that amino acids help us making the proteins. Now this actually led to the discovery and the research on the genetic codes, right? To determine the biochemical nature of the genetic code and the structure of DNA, the proportion and the deciphering, right, to understand the genetic codes was most challenging task. For this, lots people from all the different scientific backgrounds came together. It was George Gamow, right, a physicist. He argued that there are only 4 bases. He said कि सिर्फ 4 bases हैं nitrogenous bases. If they have to make a code for 20 amino acid, the combination will be 4 cube, 4 into 4 into 4. Clear? That means that we will have how many? 64 codons. Everyone, are we clear? So it was George Gamow, right? He said it कि हमें पता है कि 4 nitrogenous bases हैं और 20 amino acids हैं. Combination से देखें तो हमारे पास में 64 codons होंगे जो amino acids के लिए code करेंगे. This is important. Providing the proof that the codons were triplet was a most daunting task. We have Harbint Khurana, right? He used a chemical method. This is important. कौन सा method use किया? A chemical method. Which was a chemical method? In synthesizing RNA molecule with defined combination of bases, homopolymer and copolymer. Then we have one more scientist, Marshall. He made a cell-free system of proof for the protein synthesis. Finally helped the codes to be coded, right? To understand the codes. Then we have several enzyme, right? Polynucleotide polymerase. Remember this enzyme, everyone. कौन सा enzyme था? Polynucleotide polymerase. This was really very helpful in polymerizing RNA with defined sequence in template independent manner. This is a very, very important question, everyone, that can come. Finally, of course, we have the presenting you the genetic codes. So, now what is important for us to understand कि this first we have this 3 positions over here, right? On the 3 sides of this box, right? We have written U, C, A, G, and from one from here, one from here, one from here. This is how the codons are being made. Clear? Written, sorry, not made, but written over here. Clear, everyone? Are we clear? See, in the class only we have discussed so many, right? All of us should have the understanding of the stop codon, right? We have one more stop codon. Where is it? Yes. We should all have the understanding of the initiate codon, UAG, right? We have this over here. Then, of course, we should have an understanding of what was the first codon, right? We have it over here, U over here. So remember these things, everyone, and what will happen when you start recalling these things again and again, especially the ones which are mentioned in our NCERT, becomes super important. Clear, everyone? Yes? All of you, are we clear? Good. Now let's quickly recall the silent features of the genetic codes that we have. First, the codon is in triplet form. 61 codons are there, which codes for amino acid. 3 codons are there. They do not code for any of the amino acid, but they function as the stop codon. Some of the amino acids code more than one codon, and hence we call it, the code is the degenerate. This word was there in our slides also. The code is read in mRNA in a continuous fashion. Continuously, there's no punctuation, there's no gap. The code is nearly universal. We have the 2 exceptions over here, right, which they have mentioned, right? This is the universal code that we have, one of the example we have, and some exception we have that we have in the mitochondrial codon. Clear? Then UAG has a dual function, code for the amino acid and is the initiator codon also. And we have the list of the terminator codon. UAA, UAG, UGA. Clear? So, this is what we have studied in the genetic code, right? This part has this information, and now we are clear that how the scientists came to a discovery of that the codons are in triplet form, right? We have 4 important scientists over here, and we have 6 important silent features. And that's it. We just have this in the genetic code. Everyone, are we clear? Hello everyone, are we clear with this? Yes. Tripta, Shalini, Megdha, Radhika, Balraj. Awesome. Chaliye. Now that we are clear with this everyone, थोड़ा सा आगे बढ़ते हैं, right? अब हम देखते हैं कि how this mRNA will be reading the codes. Right? Let's take a look over here. Rule number 1. What is the rule number 1? mRNA sequence is always read starting from UAG. We know that UAG is a initiator codon. So, wherever we have the UAG, right, over here, it will always read in a triplet starting from UAG. So, this is how the initiation will start. Right? The way of dividing the nucleotide in a non-overlapping triplet is by reading framework. So, we have this 3. Where we have UAG together, that will become a part of, right, and we will consider it as an initiator codon. Clear everyone? Hello Sania, welcome to the channel and welcome to the class. Chaliye Shalini, very good. Rule number 2. What is rule number 2 everyone? Rule number 2 क्या बोलता है? Rule number 2 ये बोल रहा है कि the reading sequence, right, is always from 5' to 3'. Always the reading that will happens, right? mRNA sequence is always read from the 5' to the 3'. Clear? Okay. Now, these are the 2 important rules. First, wherever we have AUG, that is the initiator codon, and we will be reading the mRNA sequence from the 5' to the 3' end. Okay. Now, अब हम देखते हैं that what helps in converting the RNA to protein using the genetic code. अब this is important everyone. Now, we will be continuing this part in our next class, which is on the translation. But what we have today, we will be understanding कि how ये जो mRNA है, जो हमारे पास में है, which we got after the transcription, how this mRNA will be getting converted to protein using the genetic codes that we have just now discussed. So, now can you quickly recall about the tRNA? Right, tRNA है. Very, very important structure. It is a carrier adapter molecule भी बोलते हैं. It's a very important carrier for the amino acids. We call it as an adapter molecule also. Right? So this plays a super important role. We'll be discussing about the tRNA. We know that it's a carrier. It carries the correct amino acid. Smallest among the different types of RNA we have mRNA, RNA, and then we have the tRNA, which is the smallest. Previously known as the sRNA or the soluble RNA, it was soluble in 1 ml of NaCl. Now, if you take a look at the structure, right, here we have the chotu sa pyara sa our tRNA. Now, what we have in this particular structure, first, we have 5' end and this is a 3'. So, the starting is from here and the ending is over here. At the end, at the 3' end, what we have the amino acid binding site. So this is very clear for all of us to remember. Amino acid कहाँ पे attach होंगे? End पे. ऐसे बोल सकते हैं हम, right? कहाँ पे attach हो रहे हैं? At the 3' end. Clear? 5 इज अ बिगिनिंग, हम 5 से 3 डायरेक्शन में इसको रीड करते हैं, राइट, बट हमारे अमाइनो एसिड कहाँ पे अटैच हो रहे हैं, एट द 3 डैश, एंड रिमेंबर दिस एवरीवन, वी हैव अ डी आर्म, राइट, विच इज डी लूप, फिर हमारे पास में एंटी कोडॉन लूप जहां पे हमारे पास में कोडॉन्स है, राइट, एंटी कोडॉन से टू दी कोडॉन्स के ऑपोजिट है, एंड देन वी हैव दिस, सो वी हैव 1 2 3 एंड 4 ऐसे हैं, क्लियर, 4 लूप्स एब्सोल्युटली, 1 2 3 and 4, okay, 4 loops, yes, विश्वजीत, हाँ बच्चे, टीआरएनए का स्ट्रक्चर हम बात कर रहे हैं, कैरियर, ये कैरी करते हैं करेक्ट अमाइनो एसिड, राइट, और बहुत जरूरी है, इनको अडाप्टर मॉलिक्यूल भी बोलते हैं, इनका स्ट्रक्चर ऐसा दिखता है, राइट, एंड ऑफ कोर्स वी कॉल एज अ क्लोव लीफ स्ट्रक्चर, एंड ऑफ कोर्स इट्स, इफ रियलिटी में if we do the analysis, it's look like a inverted L, ऐसा लगता है, clear. 3 dash end पे we have a very important site which is the amino acid binding site. So at the 3 dash we will see that amino acid will come and attach. On the 5 dash end we don't have any attachment. Over here at the second part, right, in the lower arm we have the anticodon arm, right. फिर यहाँ पे we have one loop and we have D and T loop. Done everyone? Are we clear with this? Done? Okay? Yes. सुदेशना, सुचिता, Tripta, Shalini, Bispajit, awesome. चलिए, now that we are clear with this, let's take a look over here. अब ये थोड़ा सा स्ट्रक्चर है. Yes, I don't come live every day. We have classes on Monday, Wednesday and Thursday, 7 p.m. पे. Okay, चलिए everyone. So what we have? We have the inverted L-shaped structure. In 3D image, this is the structure of the tRNA. Clear everyone? Right? See over here, we have the binding site at 3 dash end, the connecting link and the anticodon arm. Here we have the codon and we have the anticodon. Now this is very important everyone. We know that codon is what? Codon are present in the genetic code wala usme. Clear? Yes? Anticodon, which is opposite to it, is present in the tRNA. Proteins कौन बना रहा है? Codons. Clear? Right? ये codon जो कहाँ पे है? They are actually following the, जो हमारे पास में genetic codes का जो पूरा सा checker box है वहाँ पे. What tRNA have? tRNA have the anticodon. Everyone, are we clear? There is codon and there is anticodon. So on the mRNA, what we have? On the mRNA, we have this sequence. That part is the codon. The tRNA, which will be attaching, जहाँ पे हमारे अमाइनो एसिड अटैच होंगे, उनके पास में एक एंटीकोडॉन है जो एक्चुअली मतलब कॉम्प्लीमेंट्री है बेसिकली इसके. Everyone, are we clear? But it's not the same. Clear everyone? Quick thumbs up if you are clear with this. Good. So this part में we usually have the confusion कि मैम जो जेनेटिक कोड का जो चेकर बॉक्स है वो कहाँ पे है, राइट, वो इधर उसे जो उसके जो कोड्स है वो कौन फॉलो कर रहा है, इधर दी mRNA is doing it or the tRNA. Now we have the clarity that those codes are over here. Now here we have everyone, see over here, see U A U G. Clear? Clear, very good. अगर अब ये समझ में आ गया तो एक छोटा सा पोर्शन भी अब पढ़ते हैं. Yes. Everyone, are we clear with this? चलिए, now that we are clear with this, let's talk about the tRNA charging. Now, ये हम और डिटेल में पढ़ेंगे when we are discussing about the translation also, but let's understand a small amount of it. Activation of the amino acid. Now we know that the amino acid are freely there once they are out, right? They are in the cytoplasm. So we have free amino acid in the cytoplasm. Now we have to collect the amino acid and then we have to make the proteins. So this free amino acid which is there, which is completely inactive, they're inactive. Activation happens in the presence of a very important enzyme which is the amylase synthetase and ATP. Two important things are there. We have an enzyme and we have ATP, the energy currency of the cell. Clear? Now what we will see that we have this freely amino acid which is moving in the cytoplasm. Then we have ATP and we have the enzyme. So as soon as they'll get attached, right, we will see that it requires the energy. Energy ke bina, there's no charging, right? So what will happen? These two phosphate group will say bye-bye, right? And this will become AMP. Monophosphate. We'll just have one phosphate at that particular time. So there's a release, right? We will have the amino acid which is attached to the enzyme. We will have the AMP, right? Adenosine triphosphates, and now it's adenosine monophosphate, and then we have the enzyme. This complex altogether is called as aminolase adenylate synthetase complex. This whole complex. Clear, everyone? Yes, Biswajit, I'll answer your question. Dhanya Lakshmi, I'll answer your question. Everyone, are we clear with this? All of you, are we clear with this? Clear, everyone? Yes? Okay. Now that we are clear with this, let's quickly recall activation of the amino acid. 3 amino acids are there in the cytoplasm. They are inactive. We need the enzyme and the energy. tRNA ki charging mein amino acid, tRNA without the amino acid is called as the uncharged, and as the amino acid are added, it is called the charged. So we have the active amino acid over here. We have uncharged, and when we add this, we call it as the active amino acid. See over here, when this amino acid get attached to the tRNA, it's become active. Clear? Now, here everyone, we have the charged tRNA. Are we clear up to here? Yes, are we clear up to here? Good. Now, the question that you all asked, Biswajit I think have asked, ma'am, from the DNA to the mRNA, right, you were talking about the template strand, right? Which direction? Can anyone tell me what was the direction? Do you remember, right, in the transcription we have 2 strands. How many strands we have? We have 2 strands. We have one template strand and one non-template strand. We used to call the coding strand and non-coding strand. The RNA polymerase enzyme used to work only in 5' to 3' direction. All of you remember this? Jaldi se. I'm sure you remember this, all of you. So, in which direction we'll see this happening? 5' to 3. Right, we'll remember this? Good. Very good, everyone. Very good. We'll remember like this. And the other question, I'm sorry, I forgot. Dhanya Lakshmi was asking me, what about the coding strand and the template strand, bache? The coding strand and the template strand, we had discussed this in the transcription. Now this step, right, the translation wala step is next after the transcription. So if we have to think that once, of course, we have the DNA, of course, the whole strand will slowly open, and whichever needs to be, replicated, that strand as open hoga, Y for replication will occur, and of course, winding will happen. Then from this we'll have the mRNA, right? Now in this what we have one strand that will allow the RNA polymerase to copy itself, right? Now what will happen over here? A, T, C, G, right? In this mRNA strand will have the replacement. Instead of T, what will have? We will have the U. And from this, what we'll get? We will get the proteins or the amino acid basically. So the strand, right, the DNA ka strand that are actually helping in the copying, that is called as template strand. Non-coding is the one which is the coding one is the opposite one. Clear? Clear, everyone? Good. Everyone, are we clear up to here? Yes, Radhika. Yes, Loveleen. Yes, Shalini, Meghda, Suchita. Very good. Just in case if you have any doubt, please make sure you watch the previous video which we have on the channel, which talks about the transcription and DNA replication also. Yes, everyone, are we clear with this? Thank you so much. Okay, everyone. So with this, we are done with our today's topic, that is genetic code, right? Now we will be coming live again. I'll be coming live again and we'll be discussing about the translation and the lac operon gene expression. We'll discuss about the gene regulation on Wednesday. So please make sure everyone you join the class, right? After this class, please make sure you read the NCERT once again by yourself. Okay? Read the NCERT thoroughly. Mark the important line just in case if you have any doubt. Please come back here. वापस आइए, वीडियो देखिए and वापस जाके NCERT पढ़िए. We will be doing the questions in the upcoming classes, so don't worry about the question. First, let's understand the concept thoroughly and फिर हम questions करेंगे. Clear? So with this, everyone, I will be ending the class. I hope that all of you have enjoyed the class. Yes? Please make sure everyone to hit the like button for the video. If you're new here, do take a moment and subscribe to the channel. Yes. Okay, Biswajit is asking, ma'am, why was the direction of DNA runs for anti-parallel? But DNA, I didn't get the question. We know that DNA have two strands and they are anti-parallel. Acha, you are asking why this is the case? Because, see, they cannot. In the anti-parallel, why they have anti-parallel is because of the structure also, right? So we know that we have sugar and phosphate, sugar end, free end, right? So we have 3' free end over here and over here we have the 5' free. Because of this anti-polarity, right, the anti-parallel nature, we have a stable DNA structure. If they are 5' to 5', probably it will not be a stable structure. This is all because of the structure of the DNA, the presence of the sugar, phosphate and the nitrogenous bases and how they have formed the bonds with each other. That is the reason we have this anti-polarity. Clear, Biswajit? I think if you will read the DNA replication part, you will be able to get it, right? The characteristic features of the structure of DNA, right? If you read that part, you will have the understanding, bache. Okay, everyone. So I'll end the session. Thank you so much, everyone, for coming to the class. Please make sure you read the NCERT, right? NCERT पढ़ोगे तो एकदम जोश आएगा, समझ में आएगा पूरा, and बहुत मजे आएंगे. We know that I'll talk about myself. I know that I'm not very. We just have 3 classes. No, the regularly classes नहीं हो पा रही हैं. But in the future, everyone, we are trying to have a timetable so that you all know कि अच्छा इस दिन ये हो रहा है, and we will come with these planned session, and that will be helping you. Till that, everyone, we really need your support. Please make sure you watch all the sessions that are happening on the channel. Mrinal sir, Abhishek sir, Ashish sir, Pushpendu sir, all the teachers are trying their best to make sure to give you the best quality education. So just for some more time, just bear with us. We will be coming up with a timetable and with a solid plan, and that will be helping all of us. Okay? Yes, everyone. On that note, I'll say bye-bye. Lots of love to each one of you. Please make sure you utilize this week. It's just the Monday, right? You have so many things that will be happening throughout the week. Daily थोड़ा-थोड़ा पढ़ाई करो, डरने की ज़रूरत नहीं है. Small steps at one time. And stay with us, and we'll try our best to give you the best learning experience. Okay? On that note, everyone, bye-bye, good night, and keep on learning with Akash Byju's. Lots of love, hearts. Bye-bye, everyone.